Hello, and welcome to Neurology and Neurological Basis of Speech, Language, and Hearing. In this first section, we are going to look at the neural structure and the neural transmission. The idea is to begin with the cellular level and then move into structures, later. So this lecture is going to speak about the structure of neurons and a little bit about the transmission. The transmission piece will follow in the next section. I wanted to make the videos short as possible so that they can load easily, basically. So it is important for you to view the lectures before you come to class, make notes, and identify any areas that you need explanations or you need more information about, and then come to class so that we can free the time in the class to go deeper into the material and to discuss and analyze and make connections among the concepts that we will study. So, looking at the nervous system, I understand that everyone in the class has a little background on neurology, but this class basically based on your anatomy class. But in this class, because it's dedicated completely to neuroanatomy and neurophysiology, we are going to go deeper, and we have a really, really deep and thorough understanding of the nervous system and how it functions, especially as it pertains to speech, language, and hearing. So, looking at the nervous system in general, there are two kinds of cells that make the nervous system, two categories. One category is called a neuron, and all of us know what a neuron is. And the other category is called neuroglia, or glial cells. If you think when you order a book, for example, from Amazon or somewhere else, or a piece of equipment, it usually comes in little styrofoam peanuts and packaging material that wraps the thing so it can remain intact until it reaches you. The neuroglial cells play numerous functions. Some of these functions are to actually cushion and fill the space around the neurons. But they're not just space fillers. They perform numerous critical functions, and neurons can never, ever function without the help of the neuroglia. For every neuron, there are 10 to 50 neuroglia. In other words, neuroglia or glial cells outnumber neurons by 10 to 50 times. So, we are going to discuss more of the functions of neuroglia, but I'll show you some kinds of neuroglia that are critical, and that for the central nervous system, for example. You see one here. Of course, you have a neuron and another neuron and an axon of another neuron passing away. Now you have 3 neurons in the vicinity. Now look at the myelin sheath of this neuron here on the left. You have a segment of myelin, which is a little sheet of fat that is wrapped around a segment of the axon. And each segment is separated from the next segment by one bare spot, one node, which is called the node of Ranvier. The node of Ranvier. So that basically is the area of the axon that is not myelinated. It is not insulated. It comes in contact directly with the fluid that is outside of the neuron. And I will explain later why this area is very, very important and why the firing actually occurs. It goes through these areas. So, you see the next neuron also has myelin, but you see one cell that is called the oligodendrocyte. Cyte means cell, and oligodendrocyte is a cell that is basically, it has a certain kind of sugar that makes it. But the important thing is it has many arms, as you could see, and each arm has almost like a hand like this. It has on one end, it has a sheet of myelin that wraps around an axon. So this oligodendrocyte, as you could see, myelinates various kinds of axons. So in this case, it is present inside of the brain and the central nervous system, the brain and the spinal cord. So again, it has many arms, and it makes multiple segments on different neurons at the same time, as you could see. So this is the oligodendrocyte. This is an oligodendrocyte. And on the left, you see another cell that's called the astrocyte. And so again, cyte means cell. Astro is star. So it's a cell shaped like a star. And you could see a blood vessel nearby. And this cell has branches. And each branch on its end has a little pod. Pod means foot, like podiatrist. So these pods sit on the blood vessel and has gates between it and the blood vessel. And these gates are basically, they control what goes into the nervous system. That comes out of the blood goes into the nervous system. So that is called the blood-brain barrier. And the astrocyte is the cell that makes the blood-brain barrier to allow only certain chemicals, certain substances to go into the nervous system, into the neurons. On the other side, so on one side it has these little pods docking on, connecting with the blood vessel. On the other side, it has also pods connecting onto a neuron. So what it does, it gets the food that the neuron needs, just exactly what it needs, glucose and other things, and it passes them on to the neuron. So it nourishes, it feeds the neuron. So this is one kind of glial cell. And again, the oligodendrocyte makes the myelin sheath or segments of cells inside of the brain and the spinal cord, inside of the central nervous system. So overall, we're going to speak more about the neuroglia in a minute, but overall, there are some basic functions of the neuroglia. Number one, they provide neurons with nourishment like the astrocytes, and they form the brain-blood barrier. Two, they insulate neurons, the axons of neurons. Typically, when you have a current, an electric current traveling from one place to another, the distance is important. And the longer the distance is, the more likely that some of the signal is going to be lost. So insulation is going to make the transmission of information faster and more efficient. And also it preserves a lot of the energy so it doesn't get wasted. So myelin, we cannot live without it. It is critical. So myelin is made by neuroglia, by two kinds of glial cells. Now we know one of them. We know the oligodendrocyte. The second one, Schwann cell, we'll speak about it in a minute. So another function is that some neuroglia regulate the fluids outside of the neuron, because the neuron, as we are gonna understand, is going to be very, very much influenced by the chemicals outside of its membrane, and you will see that this is basically the mechanism to channel information. So some kinds of neuroglia, they form little cylinders, imagine this is a cylinder, for example, and you get it and connect it with another, and you make a long tube, you make a hose. So these neuroglia, this kind of neuroglia, it makes these hoses, and they are used to guide brain cells, newly born brain cells, from the place that they are born. I hope that you remember from your previous classes that the place where neurons are born is called the neural crest or the neural tube. And in development, 250,000 per minute of neurons are produced and shipped out to fill different parts and to go to different parts of the nervous system. So they have to migrate to their destination. These microglial cells, these that make tubes, ropes and like hoses, they guide the migrating neurons from their site to the destination. So the neuron clings on it, and then on the process, as it keeps moving to its destination, it is fed. It gets food from that hose, from these cells, because they have nourishment inside. And by the time the neuron reaches its destination to become part of the frontal lobe or temporal, whatever, it has matured along the journey, and then it can be embedded and it stays where it belongs. So without that process, without these neuroglia, the process of cell migration during development would be impossible. And if there is a tiny problem with cell migration, the whole fetus could basically be so disordered that the nervous system could basically trigger the immune system to basically discharge that fetus out, treat it as a foreign body, as an invader, and we call this spontaneous abortion. And many of these happen in the first 2 weeks or 3 weeks of pregnancy. They are not even noticed because so again, if anything happens to disrupt cell migration during development or during pregnancy, that is going to have a devastating impact on the fetus, and if the fetus is born, the baby is going to have many problems. So also, as I mentioned, neuroglia, categories of them, the oligodendrocytes and the Schwann cells, myelinate the axons of neurons. And some neuroglial cells actually transmit signals. In addition, neuroglia or glial cells again hold neurons in their place, cushion them, and maintain them, and also some of them act like they're called microglia, like part of the immune system. They act as little white blood cells that maintain the health and the cleanliness and health of the brain and nervous system in general by cleaning out debris, by breaking down substances that are not good, and basically maintaining the health of the entire central nervous system. So these neuroglia are really, really, really critical. So there are different kinds of neurons. As you could see, you could look at the shapes and like motor neurons, pyramidal neurons, all different kinds of neurons. However, they all share a basic structure. Some components they all have, and they also transmit signals in the same way. So when we study the structure of a neuron, we look at the structure of basically any kind of neuron, because based on what neurons look like and how they function. So the basic structure of the neuron, as you are aware of it, include the cell body. Cell body of any cell is called the soma. So the term somatic comes from that. And then the dendrites, sometimes also called the dendria, and the axon hillock, and then the axon. And at the end of an axon, you have branches called synaptic branches. And at the very end of a synaptic branch, you have a little foot-like structure called pod, and that's something. So that is this little end pod, sometimes called botons as well, synapse with a dendrite of another cell to make a synapse. So let's look at some of these structures and see exactly how they work. So here we have the cell body, and the cell body contains the most important parts of the neuron. Cell body, the most important part of any cell is the nucleus. You know why? If you thought because it houses the DNA, then you are right. So the nucleus of a cell holds the information that makes the entire cell that can reproduce itself, the DNA in a cell. It is the recipe to make the entire body. This is what a DNA is. It is a recipe to construct or to build an entire body. Imagine that, say for example, you create a map, and that map is self-regulating. Somehow you just write it down, program it, put it into a computer chip, and then take it and throw it into the ground, and then somehow it activates itself and it starts to build you a building. Imagine something like that. I mean, we see it every day, but we don't even really think about it this way. Imagine the seed, a little seed, any kind of seed, and how it has the map of a tree and the description and everything of a tree, and then it is all in the DNA. You just throw it into the ground, give it the right conditions, and then it starts to build itself out of the ground, and it will be a gigantic tree. So the DNA is, again, the map to build a whole body or to build body parts on different other cells. When an action potential comes in and say something that stimulates the neuron, it goes into this nucleus to be processed. And if it is significant and you need to respond to it, the nucleus is going to produce, to copy certain genes are going to be copied so that you can produce certain chemicals, so that you can make certain mechanism like neurotransmission components, protein channels, whatever, and ship them out to different parts to maintain the neuron and make it able to perform its function. So the cell body also has many little organs called organelles. So the nucleus is the biggest, or kind of organ cell body, but other areas that have ribosomes and different kinds of compartments, these are called organelles. So the body of the cell contains its most of its organelles. It contains its nucleus. On the different parts of the body, there are little branches called dendrites. Dendron means tree, and dendrite is basically these little branches. There are hundreds and thousands of them. They go out and branch out. So these are called dendrites. Dendrites are the only part of a neuron that can receive cells in addition to the cell body itself. So the cell body and dendrites are the two single parts of a neuron that can receive signals. Just to understand and remind you, the signal in a neuron travels one-way direction. It doesn't go back. It travels only in one direction. It comes from the dendrites and goes into the cell body. From the cell body, it goes to the axon hillock, and then through the axon, to the axon terminals, and then to another neuron. So then now we see the axon, and the axon has little segments of myelin that are made out of either oligodendrocyte cells, if this is a central nervous system neuron, or Schwann cells, if that is a peripheral nervous system that is out of the cranial nerves and the spinal nerves. So they have this kind of myelin that is made out of Schwann cells. I'll explain the difference again, but what you see in front of you, the model that we are using here, is basically the motor neuron model, and it is similar to many other neurons, but this is the typical one that basically we use as a model. So what you see in front of you now, if I'm saying it's a motor neuron, this is a kind of neuron that exists in one part of the peripheral nervous system, because it has myelin segments made out of Schwann cells. Every little segment here is a Schwann cell. So the Schwann cell is basically a cell that is like a sheet of fat that grows. As it grows as a little piece, wraps itself around a segment of the neuron, and as with development it adds more layers, and it continues to add more layers, and it continues to add more layers, and so on. It can reach up to 16 layers each segment. What a baby can do from birth and on is like movements and stuff is determined by these, by how many layers of myelin there are. The frontal part of the body, the brain, called neocortex or the prefrontal cortex. Myelination in it continues in the 30s. So it is continuing to mature through the 30s, like 36, 38. And I'm kind of highlighting the importance of myelin for the function of the nervous system. But what you see in front of you are each single segment of these that you see is one single Schwann cell. Now, do you understand the difference between a Schwann cell and an oligodendrocyte? We know they are similar in the fact that both of them are a kind of neuroglial cell. However, one functions in the central nervous system, one functions in the peripheral nervous system. In addition, oligodendrocytes have different branches. They can myelinate different segments, more than one segment of the same neuron, and in addition to that, they also myelinate different neurons, multiple, hundreds of other neurons. A Schwann cell is only one single sheet that wraps itself to make a single segment, and each segment, each Schwann cell is separated from the other Schwann cell by the node of Ranvier, as you could see. The oligodendrocyte myelin also has nodes of Ranvier. So every segment is separated from the next one by a little uncovered, uninsulated spot that is directly exposed to the fluid, and I'll explain to you why this is important. So then we have the synaptic branches. A branch is the whole branch, and at the very end we have these what are called synaptic terminals or synaptic pods. So either way. And the synaptic terminals, synapse, they get in contact with dendrites from another cell so that they make a synapse and can share information. Okay. Sorry, it froze for a minute. So now the cell body, in addition, I explained, the cell body has little particular sites where it actually, you have synaptic terminals from another neuron coming and making contact, making a synapse with the cell body itself, not the dendrites. So I told you that only the cell body and the dendrites are the sites for making synapses, for receiving information from other neurons. So a synaptic terminal from one neuron can come directly and go to a special site on the cell body, and then that's what makes this site special, is that it has receptor channels that can allow the flow in and out of certain chemicals that we will speak about later. So cell bodies also have little areas for that have receptor channels, and they can form synapses. If a synapse is formed between a synaptic terminal of a neuron and the body of another neuron that is going to get signals from it, that synaptic kind of synapse is called axon, or axo from axon, axosomatic. Somatic means body. It has to do with the body. Axosomatic synapse. If the so you could see here an axosomatic synapse here, where the synaptic terminal of a neuron synapses directly onto the body of the cell. When the dendrite, the axon of a neuron makes a synapse with the dendrite of another neuron. Sorry, this is so this is, here's one neuron. I'm gonna show you when they actually this area here. So you have the axon of this neuron. One axon comes, axon terminal, and contacts dendrite of this big neuron, and together they make the axodendritic synapse. So axo has to do with axon, dendritic has to do with the dendrite. So if it is between a synapse, between a synaptic terminal and a dendrite, it's called axodendritic. So I explained what the dendrites are and what they do, so let's move on. The process of development involves development of all these dendrites of neurons. And that process is called arborization. Arborization is the development of these different, all these dendrites of neurons during development. Also, dendrites have something called a dendritic spine, which when you look at it, it has like little, you know, when in the spring, when you see the budding branches before they actually become branches, these little buds, they are called dendritic spines. And these are critical in information processing, in making memory and information possible. So, as you learn, these these dendritic spines, they can be eliminated, or you could immediately start to sprout new dendritic spines. And they come in different shapes and stuff. A lot of new research now emphasizes how critical these are in shaping how we learn and how we behave. So the axon now, the axon itself is like think about it like an electric wire, or like actually it is better to describe it as an electric wire. It has insulation, it has the actual wire inside that actually carries the signal. So inside, again, you know, you have these little myelin segments separating, basically separating most of the axon from the extracellular fluid outside, insulating. And you have these gaps, which are the nodes of Ranvier. They are the only sites that have channels, receptor channels, to allow chemicals in and out. And this is why it is here that the firing occurs. So what you see here, for example, in this little neuron, you can enlarge it and look at it, is what is underneath two segments of myelin. You basically, you can see the flow goes directly flows under the Schwann cell and flows through. And then when the current reaches these gates, it depolarizes the gates, and it causes the sodium and potassium to go in and out. And that basically is the firing. And then it continues to the next. So, and then because it shows between segments, it seems as if the signal is jumping. It really isn't jumping. It just keeps flowing in that direction, and only these little areas just flash, 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 and one after the other, of course, in sequence, like dominoes. So you see it almost like jumping. They call it saltatory conduction. So again, you can see now the Schwann cell, how it is basically one single cell wraps itself around one single segment of the axon, leaving between it and the next segment, leaving the node of Ranvier, that bare area that is directly exposed to the extracellular fluid. And it is the only area that has the receptor channels. And this is why the action potential that is generated in the axon hillock here is going to be transmitted, through the axon, and when it reaches every segment, it regenerates, it triggers another action potential, and that action potential travels through and triggers another action potential, and it keeps like dominoes, like one falls and the next one and the next one and the next one. It keeps going in that direction. So you see the action only in the bare spots, which are nodes of Ranvier, but the signal itself really doesn't kind of jump. The area here that connects the body of the neuron with the axon, this triangular area here, is called the axon hillock. This is where firing takes place, and we'll explain it in detail later. So again, you could see now the oligo and oligodendrocyte. So don't think when I say the oligodendrocyte that there's one single cell. Of course, that would be really not very smart. There are millions and billions of them. So we just take one because one is like all others. So you could see it now here, an oligodendrocyte, and you can see how it myelinates multiple segments of neuron, one single neuron, and multiple neurons at the same time. So again, know the difference between the two in terms of where do oligodendrocytes myelinate and where Schwann cells myelinate, what kind of parts of the nervous system they myelinate. So now let's look at the synaptic terminal. The synaptic terminal, as I mentioned, is the very end of the synaptic branch. And it comes like a little button, or better described as the pod or foot, as you could see it now, shaped like a foot. And it is one part that is one part of the synapse itself. A synapse is the point where two neurons communicate with each other, and the synaptic complex is sometimes, when you say the synapse, it refers to the synaptic complex itself, but either one is fine. So this is the synaptic pod, because signals travel in one direction from one neuron, from the cell body of a neuron to the axon. The synaptic terminal is bringing in a signal, okay? So when you look at the space between the synaptic terminal and the part of the cell body, or the part of the dendrite that is synapsing with it, there's that synaptic cleft that is a tiny space between them. So in reality, they do not really touch. There's a tiny space in between them, and they communicate by releasing fluids and stuff. So the synaptic terminal, we describe it as part of this neuron. We say before the synapse. We call it the presynaptic terminal, or the presynaptic neuron. So that neuron is the one that is sending a signal. The presynaptic neuron is the one that sends a signal. The one that receives a signal is called the postsynaptic neuron, the one behind the synapse, behind the little gap between the two neurons. So the presynaptic terminal releases neurotransmitters into the synaptic space, or called synaptic junction, or called synaptic cleft. All of them mean the same. And that neurotransmitter finally is dumped into the synaptic cleft, and then it goes to the receptor channels, and it binds on the surface of receptor channels. Neurotransmitters do not enter into the postsynaptic neuron. They do not enter. They just go to the surface of the receptor channel and activate it to make it open to allow sodium, potassium, chloride, whatever it is, or to dock and close the gate. So it can be either excitatory signal that opens, or an excitatory neurotransmitter, or an inhibitory neurotransmitter that docks, makes the cell, prevent the cell from opening. So the axon HELOC is a critical piece of the neuron. Really, really critical. So look at this triangle here. That triangle that connects the cell body with the axon is the axon HELOC. This is where the actual firing of the neuron takes place. It relies how it fires, as we will understand later, how it fires. It relies on all the signals coming to it. If a signal is important, finally it will find its way to the axon HELOC. Once it reaches there, then, the axon HELOC gets signals now from every single dendrite, every single synapse on the body or the dendrites. Hundreds and thousands of signals. Because some of these neurons communicate with 50,000 or more. So now, some of these signals are inhibitory, to say to the axon HELOC, fire, don't fire. Some of these signals are excitatory, say, go fire. So how does it decide? And in addition, when we speak about action potentials and all this and the transmission piece, there are some little messages that are called graded potentials, that they are like little energy kind of bringing some message, but they are not strong enough. They might die down before they reach the axon HELOC. So anyways, hundreds and thousands of messages come to the axon HELOC. Some of them say, do this, and the other ones say, no, don't do that, do this instead. The axon HELOC decides based on the majority, democracy, and the majority of the signals coming in. If more signals are telling it to act and fire, it will fire. If more signals are telling it to inhibit, it will prevent the neuron from firing. It doesn't fire. And if many weak signals come together, add their energy, and they become important together, they can convince the axon HELOC to actually fire an action potential, as we'll describe later. So that area is an area where the neuron integrates its functions. It takes all kinds of information, processes, and makes a decision whether to fire or not to fire. And once it fires, it transmits the signal through the axon. And we will explain in detail, when we do transmission, we'll explain in detail how exactly this process takes place. It is very, very interesting. So the synapse. The synapse, again, is the point of contact between one neuron and another. And the synaptic complex, there are two kinds of synapses we'll talk about, but the synaptic complex includes the synaptic terminal, or the pod, and let's use my hand as the pod, and then the area of the dendrite, or the area of the body that has the channels, receptor channels, where the neurotransmitter coming from the synaptic terminal combined on the channels so that it can allow in chemicals that will transmit the signal. So first we have the synaptic terminal, or the pod, of the presynaptic neuron. Then we have the dendrite, or the part of the body that has the specialized receptor channels. These are, again, the dendrites are always, in that case, part of the postsynaptic neuron. Because what we are describing here is two neurons, one has a signal, and this is going to pass it to the other neuron. So in the space between them, that tiny space in between them is actually like 1/1000th of a meter, like really, really like very, very tiny. Is called the synaptic junction or the synaptic cleft or the synaptic space. So again, in terms of the connections that the synaptic terminals make with the other cell, with the postsynaptic neuron, whether they connect or synapse with a dendrite or synapse directly onto the body, there are two kinds, as I explained before. If they synapse, like the terminal synapses with a dendrite, we call this axodendritic, from the axon connecting with the dendrite. If it connects or synapses with the body itself, we call this axosomatic. So that's in terms of how the synapse itself is connected. In terms of function, in terms of physiology, there are two kinds. One is called the electric synapse, and one is called the chemical synapse. And they are different, but simply the greatest majority of synapses in the nervous system are of the chemical kind, chemical synapses. The electrical synapses are few, but they do exist. They exist more in certain animals than in humans. So the electrical synapse, let me show you the image first and explain. You have now a synaptic terminal coming from the presynaptic neuron. And then you have this area here, the postsynaptic neuron, whether that's part of the cell body or part of the dendrite itself. What do you notice now? You don't notice that the space between the synaptic cleft is not completely wide open. In here, protein channels from one cell connect with the protein channels of the other cell, and they make a pipe together. They are coupled as a pipe. So in effect, now you have pipes that connect the fluids inside of one neuron with the fluids of the next neighboring neuron. So you could see now, for example here, this is a gate here where the fluid can flow directly from one to the other. That is called the gap junction. And you could see it here as well. And of course there are others. So again, in the electrical neuron, the fluid and the electrical synapse, the fluid inside of a cell, a neuron, is connected directly with the fluid of the other cell. So when the signal, the electric signal comes in, it doesn't need agents. It goes directly from the cell into the other cell directly, and fast and very, very, very quick. So that allows basically powerful actions. It allows people to act very, very quickly, or animals to act on instinct, like really, really very fast. It allows neurons that function together to do a certain function to synchronize their firing so that all of them just immediately fire together. So again, this is not it is just rare in the human nervous system. And I need you to understand that in this case, the fluid of, the two membranes of the adjacent neurons are physically connected with gates, with pipes. that allow a chemical that brings a signal or an electric impulse to pass directly without any intermediary from one cell to the next. The chemical synapse, the most common one in the human nervous system now, again, the terminal comes from the presynaptic neuron. The dendrite, or the part of the body, come from the postsynaptic neuron, and the synaptic cleft or junction, synaptic cleft or the synaptic space separates them. And in that case, the neurotransmitters coming from the synaptic terminal are released in a certain way into the synaptic cleft, in a way so that the membrane of the synaptic terminal doesn't allow anything in, and it doesn't allow anything out except for the vesicles, for the what do you call them, the neurotransmitters. So in a way, as the electric signal comes through from the axon helix and finally finds its way to the axon terminal, it triggers changes in the membrane of the synaptic terminal, and that membrane is going to attract these little vesicles and pull them towards it. There are little hooks called synaptic snares that come out and then pull the bubble or the vesicle close and close and close until it fuses. So as it fuses with the membrane, it starts now to open one end, and then now you have the neurotransmitters expelled out effectively without allowing any substances in and without allowing anything out except for the neurotransmitters. And the neurotransmitter then finds a receptor gate where it acts like a key on a lock. Every neurotransmitter, has a particular spot that only that can fit. And some neurotransmitters can activate like 12 types of channels. But in every case, it's exactly like a key fits into a lock. Once it fits in, it is going to do its job by opening or closing that channel to allow ions like sodium, calcium, potassium, chloride, so on. So now let's look at the structure of receptor channels, just to give you a piece of information. The system here is very, very flexible. And as you sit and learn, things, for example, as you are exposed to demands and in a semester, for example, you have so much work and so many responsibilities and you study and learn and all of this. These protein channels or these receptor channels can decrease, become a few, or can increase. And I hope this is the case, that as you learn more, you are going to become more efficient and you get more and more and more of these protein channels or receptor channel gates, so that you also get more neurotransmitters so that you can process information really, really quickly and proficiently. So they are not stable all the time. They increase and decrease based on many other factors. So, presuming that you have had your biology class already, hopefully also your chemistry, when you look at the cell membrane of a neuron or the cell membrane of any cell, they're the same. Cell membranes of a neuron, basically made out of like little tiny very small structures, like little tails with heads like this, like a nail, imagine like a nail, of fat, lipid. And these lipids, as you could see them here if you magnify this picture, that the heads are sticking on one side, and then inside of the cell the heads are facing the inside of the cell. So on the other side, the heads are facing the outside of the cell. And because water and fat don't mix, they basically, these lipids get attracted to each other and stick together, and the other layer facing the inside of the cell also stick together, and the tails are in between them. And this is why the membrane of the cell is always flexible, like soap bubbles, very, very flexible. Now, the cell has to control its internal environment. The way it controls it is by having gates, by having channels. The channels are inserted into the membrane so that they can have openings, and these openings can allow in and out certain chemicals to enable the cell to do its function. So what you see, all these cells are made, most of these, they are called protein channels. A protein, as you could see here, is a huge, a gigantic molecule. Molecule is made out of atoms and amino acids and stuff. So the molecule, the protein, is very, very big. Imagine like taking, say, 1,000 golf balls, or 1,000 actually big balls, and stringing them together, all of them, and then coiling them on top of each other. Now you can make a huge, big structure that has a hole inside, but outside they're all the balls are lined up and strung together, and you can make different structures out of them. So this is how the protein works. The protein is the building block of the human body or the animal body. So it makes different kinds of things that we need, and in this case, a protein channel is made so that it has a particular hole inside of it, and it has a lock. Some of them have locks, imagine. And they can close, they can open, but that lock is controlled by that little neurotransmitter that comes and docks on the surface of the gate, and then it magically opens to allow certain chemicals in or out. So there are two kinds of receptor channels that we are going to look at in terms of neural transmission. These are, one is called the ligand-gated receptor channel. Ligare means to tie, ligare. So ligament is that, kind of that little thread that connects a bone, with the bone. So ligand is the thing that ties itself to, that connects onto the receptor cell. Ligand gated, so means when you have a ligand docking on top of, so the neurotransmitter is a ligand docking on the surface of the receptor channel, it is going to activate the channel or to stop it from opening, to close or open it. So in that case, we call it ligand gated. The neurotransmitter, the ligand, causes the channel to act as a gate. Instead of a pipe allowing things to flow freely in and out, no, it can act as a gate to close and to open on demand. So that's the ligand-gated receptor channel. It uses neurotransmitters all the time to open and close, and the action of the neurotransmitter immediately is once it opens the gate, it is going to allow an influx of ions or chemicals to go into the cell, or an influx inside of the cell to exit the cell, or to basically close the cell. So the neurotransmitter in the ligand-gated receptor channel immediately triggers an action potential. Once the gate opens, then the chemical composition changes inside of the cell, and you get an action potential. But there's another kind of channel called the metabotropic receptor channel. The name seems like, hard, but think of this. Metabolic, and then the tropic is an adjective. So it means it's a kind of receptor channel that involves consumption of energy, additional consumption of energy. And that kind of channel, metabotropic receptor, it is not really a protein channel. And it doesn't, even though a neurotransmitter docks on it, there's no gate to open or close. I'll explain. So the metabotropic receptor channel is part of a transduction pathway, a pathway that acts like dominoes. Once the neurotransmitter docks onto that kind of channel, it is going to cause it to trigger another protein to act, and that protein will trigger another protein to act, and then that protein is going to make a change, and then finally you deploy something that is called a second messenger, which directly goes and spreads the signal through the cell body. So again, metabotropic does not have a gate, and it acts as a part of a transduction pathway where a cascade of events, once it is set, each event will trigger the occurrence of another event until the signal gets transmitted. Again, metabotropic, they are less common than the ligand-gated receptor channels. So here, you could see now the cell membrane. You can see the heads of the lipids, the fats, how they line up together and close to prevent the water from coming in. And on the other side, you have the heads facing the other side to prevent chemicals inside of the cell body from exiting. And then you have these protein channel gates that are span across the membrane, and they can open or close. Of course, you could see this one, it is like cut so that you could see through it. But of course, it is not something that you really see through. So in that case, the ligand-gated receptor channel, again, you have a neurotransmitter that comes from the presynaptic neuron. It docks onto a special site, like a key and into a lock on top of the receptor channel. And that is going to either make it open to allow chemicals from outside to go in, or chemicals inside to go out. So that is straightforward. And so it makes it act like a gate. And a gate is to control who's going in, who's going out. The metabotropic receptor channel here, again, it doesn't act like a gate. It causes a chain reaction where, for example, once it gets the neurotransmitter binds on it, it's going to send the signal to a neighboring neuron, a neighboring protein called the G protein, and the G protein is going to activate another protein, basically. So finally, you get to produce, go through 2 steps of energy consumption, and then finally you deploy a second messenger, a second messenger inside of the cell body that basically takes that message and brings it and spreads it so quickly through the cell, because these second messengers are very, very tiny molecules. They can travel very quickly and diffuse. They can flow very quickly. And because of that, with the second messenger inside of the cell now transmits the signal, the neurotransmitter now is known as the first messenger. So the first messenger bringing a signal, and then it sets all this chain reaction, and then a second messenger takes the message and spreads it, broadcasts it through the cell until that message reaches the axon hillock. The two most common second messengers in the nervous system and the body generally are a chemical called cyclic AMP and the calcium ions, which is calcium that has missing two neurons. Missing two electrons. I'll explain that in the second segment. So the second messenger system, it has multiple steps. It is slower to start. However, once they function, once the second messenger takes that signal, it's going to activate multiple potassium channels at once to close them or to open them at once. So the action is very, very powerful immediately. So again, compared to ligand-gated receptor channels, second messenger system activity can last for minutes or even for hours. So that is going to be critical for consolidation of memories, for connecting your previous knowledge with your past knowledge, and so on. So both of them are needed, but the neurons that are going to fire for a long time are going to require second messengers. So this basically brings to conclusion the part that speaks about the structure of the neuron and some basic functions of these structures. And the next segment is going to discuss exactly the process of how to transmit signals, how we can make a neuron fire. And I do hope that you find this material accessible given your background, and that as we continue that you enjoy the class, because it is very, very exciting. It has lots and lots of things that will really make the difference between you and any grad student or any SLP. I hope that you put the amount of work that is needed into it, and I'm very excited to teach this course and to have you in the class. Thank you.